541 million years ago, something catastrophic happened. An entire ecosystem vanished. Not gradually, not through competition or climate change, but suddenly, completely, within a few million years. Forests of meter-tall organisms, gone. Fields of quilted creatures the size of car hoods, extinct. Spiral forms with impossible geometries, deleted from existence. For tens of millions of years, these organisms had dominated Earth's oceans, from shallow coasts to the deep sea, globally distributed, successful, thriving. Then they disappeared. No transitional forms, no descendants, no evolutionary legacy, as if someone reached down and erased them. But we don't call it an extinction. We call it the Cambrian explosion. We celebrate what appeared. Animals, shells, eyes, predators, the ancestors of everything alive today. We focus on the winners, and we forget the losers, the organisms that were here first, that made complex life possible, then vanished before the rules were written. They were called the Ediacarans, and their extinction might be the most important event in the history of life. Because without it, without their failure, animals would never have succeeded. We wouldn't exist. Before we go any further, please consider subscribing and liking this video. Your support truly helps us continue this work. Thank you. The story of the Ediacaran organisms doesn't begin in England in 1957. It begins 11 years earlier, halfway around the world, in the outback of South Australia. 1946. World War II had just ended. Reginald Sprigg, a young geologist working for the South Australian government, was conducting a survey of abandoned mines in the Flinders Ranges, a rugged mountain chain north of Adelaide. The government wanted to know if any of these mines could be reopened. Australia needed copper, lead, zinc, minerals to rebuild after the war. Sprigg's job was to assess the geology, take samples, file reports. It was supposed to be routine, but Sprigg was more than a mining geologist. He was curious. And one afternoon, while eating lunch on a rocky hillside overlooking the Ediacara Hills, he noticed something odd on the surface of a sandstone slab. Circular impressions, dozens of them. Some the size of coins, others as large as dinner plates. They looked like fossils. Specifically, they looked like jellyfish. Medusae, washed up on an ancient beach and preserved as the sand turned to stone. Except the rock Sprigg was sitting on was Precambrian, over 500 million years old. And jellyfish, according to the textbooks, didn't exist back then. Nothing complex did. Sprigg should have ignored the impressions, marked them down as sedimentary structures, ripples or concretions formed by water and minerals. That's what any sensible geologist would have done. But Sprigg didn't. He collected samples, dozens of slabs, and when he returned to Adelaide, he sent photographs and descriptions to paleontologists around the world. The response was underwhelming. Most ignored him. A few wrote back politely suggesting he'd misidentified the rocks, that the fossils were probably Cambrian, from a younger layer that had been mislabeled. One reviewer at the journal Nature rejected his paper outright, stating that the fossils were poorly preserved and the age determination was questionable. Even the Premier of South Australia, when asked to fund further expeditions, reportedly told Sprigg, "Give me copper, not fossils." Sprigg was devastated, but he didn't give up. He continued collecting, mapping, documenting. Over the next decade, he amassed a collection of hundreds of specimens, circular forms, frond-like shapes, segmented oval creatures, things that looked like worms, or maybe slugs, or maybe nothing anyone had ever seen before. He published his findings in obscure Australian journals, where they were largely ignored by the international scientific community. The problem was simple. Precambrian fossils weren't supposed to exist, and if they didn't fit the paradigm, they were dismissed. Then, in 1957, Roger Mason found Charnia, and suddenly, Spriggs' Australian fossils didn't seem so crazy anymore, because Charnia was unambiguous. It was clearly biological, clearly Precambrian, and it was found in England, not some remote corner of Australia that scientists could conveniently ignore. The British geological establishment couldn't dismiss it. Trevor Ford presented the findings at the Geological Society of London, and within a year, the floodgates opened. Paleontologists started looking at Precambrian rocks with fresh eyes, and they started finding fossils everywhere. In Namibia, along the Skeleton Coast, German geologists discovered beautifully preserved specimens in ancient riverbeds. In Russia, near the White Sea, Soviet scientists found dozens of new forms. In China, in Siberia, in the Canadian Arctic, even in Death Valley, California. Everywhere geologists looked, they found traces of the same mysterious organisms: fronds, discs, quilted ovals, segmented forms, all from the same narrow window of geological time, between 635 million and 541 million years ago. By the early 2000s, the evidence was overwhelming. This wasn't a fluke. It was a global ecosystem. And in 2004, the International Commission on Stratigraphy did something it hadn't done in over a century. It created a new geological period, the Ediacaran, named after the Ediacara Hills in South Australia, where Reg Sprigg first found his fossils 60 years earlier. It was the first new period added to the geological timescale since the 1860s, and it represented one of the most important intervals in the history of life on Earth. But here's what made the Ediacaran so significant. It wasn't just that these organisms existed, it was that they were huge. In a world where life had been microscopic for 3 billion years, suddenly there were creatures you could hold in your hand. Some, like Charnia, reached 60 centimeters in length. Others, like Dickinsonia, a flat, quilted oval creature, could grow up to 1.5 meters across. These weren't bacteria. They were macroscopic, multicellular, complex, and they appeared almost out of nowhere. The question was, why? What changed? What allowed life to suddenly get big? The answer lies in an event that happened just before the Ediacaran began. An event so extreme, so catastrophic, that it nearly sterilized the planet. Scientists call it Snowball Earth. Between 720 and 635 million years ago, Earth experienced a series of global glaciations. Not just ice ages, where glaciers advance and retreat from the poles. Total glaciations. The entire planet, from equator to pole, frozen solid. Oceans covered in ice up to 1 kilometer thick. Land buried under glaciers. Temperatures plunging to minus 50 degrees Celsius. For millions of years, Earth was a lifeless white ball, reflecting sunlight back into space, locked in a feedback loop of cold. How life survived Snowball Earth is still debated. Most scientists believe that volcanic activity continued, even under the ice. Volcanoes punched through the glaciers, releasing carbon dioxide into the atmosphere. And slowly, over tens of millions of years, CO2 accumulated. With no rain to wash it out, no weathering to remove it, greenhouse gases built up until the planet finally, catastrophically, warmed. The ice melted, oceans thawed, and Earth swung from frozen wasteland to hothouse in less than 1,000 years. The aftermath was chaos. Meltwater flooded the oceans, carrying nutrients, minerals, and organic carbon that had been locked in ice for millions of years. The oceans, starved of life during Snowball Earth, suddenly became incredibly fertile. And life, which had clung to existence in a few volcanic refuges, exploded. This was the environment that gave rise to the Ediacaran organisms, a world recovering from catastrophe, a world rich in nutrients, low in competition, and, crucially, a world with rising oxygen levels. For most of Earth's history, the atmosphere contained almost no free oxygen. Early life was anaerobic. It didn't need oxygen. In fact, oxygen was poison. But around 2.4 billion years ago, cyanobacteria evolved photosynthesis and began producing oxygen as a waste product. For the next 2 billion years, this oxygen was absorbed by iron in the oceans, forming rust. Banded iron formations, the red and black striped rocks common in ancient shields. But eventually, the iron was used up, and oxygen began accumulating in the atmosphere. By the end of Snowball Earth, oxygen levels had reached perhaps 10% of modern levels. Still low by today's standards, but enough. Enough for cells to generate energy more efficiently through aerobic respiration. Enough to support larger, more complex organisms. Enough to allow multicellularity. And so, in the quiet oceans of the early Ediacaran, something extraordinary happened. Life got big. But these weren't animals. Not yet. They were something else. And understanding what they were requires going to one of the most important fossil sites on Earth. A windswept peninsula on the eastern coast of Newfoundland, Canada. A place called Mistaken Point. Mistaken Point is not an easy place to reach. It's at the southeastern tip of the Avalon Peninsula, hours from the nearest town, accessible only by rough dirt roads that turn to mud in the rain. The coastline is treacherous. Sheer cliffs drop 50 meters to the North Atlantic, waves crashing against rocks that have seen 400 million years of storms. Fog rolls in without warning, thick and disorienting. Early sailors mistook this point for the entrance to a safe harbor, only to find themselves smashed against the cliffs, hence the name. But for paleontologists, Mistaken Point is paradise. Because here, on broad, flat bedding plains exposed by erosion, are some of the best preserved Ediacaran fossils in the world. Not just individual specimens, but entire communities. Hundreds of organisms frozen in a place exactly as they lived 565 million years ago. It's like a snapshot, a photograph from deep time. The fossils at Mistaken Point are different from those Reg Sprig found in Australia. They're not impressions in sandstone. They're casts. Three-dimensional molds preserved in volcanic ash. Here's what happened. These organisms were living on the seafloor, perhaps 1,000 meters deep, far below the reach of sunlight. Then, without warning, a nearby volcano erupted. A submarine pyroclastic flow, a dense cloud of superheated ash and gas, swept across the ocean floor, burying everything in its path. Death was instantaneous. The ash hardened into a fine-grained rock called tuff, and the organisms, smothered but undecayed, left perfect molds of their bodies. What those molds reveal is astonishing. There are fronds, dozens of species of fronds, ranging from a few centimeters to over a meter tall, anchored to the seafloor by a disc-shaped holdfast. Some, like Charnia, have a central stem with branches splitting off in a regular fractal pattern. Others, like Fractofusus, have a more chaotic branching structure. Still others, like Bradgatia, resemble feathers or ferns, with hundreds of tiny branches radiating from a central axis. And they're not randomly scattered, they're organized, spaced out in patterns that suggest competition for resources. Larger fronds surrounded by smaller ones, like trees in a forest with understory plants filling the gaps. Some species seem to cluster together, while others are solitary. There's structure here, ecology. These weren't just organisms, they were communities. But what were they? That's the question that has consumed paleontologists for 60 years. And the answer is far from clear. In the 1960s and 70s, when Ediacaran fossils were first being studied in detail, the dominant hypothesis was proposed by a South Australian paleontologist named Martin Gleisner. Gleisner looked at the fossils and saw animals. He compared Charnia to modern sea pens, soft-bodied cnidarians related to corals and jellyfish. He compared the disc-shaped fossils, like Cyclomedusa, to jellyfish. He compared segmented oval forms, like Sprigina, to annelid worms or early arthropods. And the strange three-lobed creature called Tribrachidium he thought might be an early echinoderm, related to starfish and sea urchins. Gleisner's interpretations were widely accepted. Museums built dioramas showing Ediacaran oceans filled with jellyfish, worms, and sea pens, swaying in gentle currents. It was comforting, familiar. These were the ancestors of modern animals, the argument went, just more primitive. The Ediacaran fauna represented the roots of the animal tree, the first branches before the explosion of diversity in the Cambrian. But there were problems, serious problems. And by the 1980s, cracks were beginning to show in Gleisner's hypothesis. The first problem was growth. Modern sea pens grow from the bottom up. They have a bulbous base, an anchor embedded in the sediment, and the frond grows upward from there. But Charnia grows from the top down. The oldest, most mature branches are at the base. The youngest, smallest branches are at the tip. It's completely backwards. And more importantly, Charnia doesn't have an anchor structure. It has a flat disc sitting on the surface of the sediment, not penetrating it. Sea pens can retract into their burrows when threatened. Charnia couldn't. It was stuck, immobile, vulnerable. The second problem was the discs. Cyclomedusa, the supposed jellyfish, doesn't look anything like a jellyfish when you examine it closely. Jellyfish are radially symmetrical, yes, but they have tentacles, gonads, a stomach, some kind of internal structure. Cyclomedusa is just concentric circles, rings, like a target, no tentacles, no evidence of a mouth or any feeding apparatus. And it wasn't free swimming. The fossils are always found lying flat on the seafloor, sometimes with a holdfast structure suggesting they were attached. Jellyfish drift. Cyclomedusa didn't. The third problem was symmetry. Many Ediacaran organisms exhibit a type of symmetry called glide reflection, or glide plane symmetry. This is a geometric transformation where you reflect an object across a line and then translate it. The result is a pattern that looks almost mirror symmetrical, but slightly offset. It's common in textiles, in quilts and woven patterns, but it's unknown in animals. Animals use radial symmetry, like jellyfish and starfish, or bilateral symmetry, like worms and humans. They don't use glide plane symmetry. It's geometrically possible, but biology has never found a use for it, except in the Ediacaran. And the fourth problem, the biggest problem, was the complete absence of any internal anatomy. When you look at well-preserved Ediacaran fossils under a microscope, when you examine them with CT scanners and laser imaging, you find nothing. No organs, no digestive tract, no circulatory system, no muscles, no nervous tissue, just external surfaces, quilted patterns, textures, but nothing inside. It's as if these organisms were empty, hollow, inflated bags of tissue with no internal complexity whatsoever. So if they weren't animals, what were they? In the late 1980s, a German paleontologist named Adolf Seilacher proposed a radical alternative. Seilacher had spent decades studying Ediacaran fossils from Namibia, some of the best preserved specimens in the world. And he concluded that Glasner was wrong. These organisms weren't animals. They weren't plants. They weren't fungi. They were something else entirely. A separate kingdom of life that evolved, thrived for tens of millions of years, and then went extinct, leaving no descendants. Seilacher called them Vendobionta, named after the Vendian, an old Russian term for the late Precambrian. And he argued that they had a unique body plan, unlike anything alive today. He called it the quilted construction. Imagine a mattress, an air mattress specifically, made up of multiple inflatable chambers, each separated by a thin wall. Now imagine that mattress is the organism. The chambers are filled with fluid or gas, giving the organism structure and rigidity without needing a skeleton. The walls between chambers are thin membranes, permeable, allowing gas exchange. The entire organism is flat, maximizing surface area relative to volume, allowing it to absorb nutrients directly from the water through its skin. This is osmotrophy, the same feeding strategy used by fungi. Fungi don't have mouths. They secrete enzymes into their environment, breaking down organic matter externally, and then absorb the resulting nutrients through their cell walls. Seilacher proposed that Ediacaran organisms did something similar. They lay flat on the seafloor, absorbing dissolved organic carbon directly from the water. No mouth, no gut, no anus, just diffusion across a membrane. It was a brilliant, audacious hypothesis, and it explained a lot. It explained the lack of internal organs. It explained the quilted texture. It explained why these organisms were so flat, why they had such high surface area to volume ratios. It even explained the strange symmetries. Because if you're building an organism out of modular, repeating units, inflatable chambers arranged in a fractal or quilted pattern, you're not constrained by the same rules that govern animals. You can experiment with geometries that animal body plans can't achieve. But Seilacher's hypothesis also raised enormous questions. If Ediacaran organisms absorbed nutrients through their skin, they needed to live in water rich with dissolved organic carbon. But where did that carbon come from? In modern oceans, dissolved organic carbon comes from decaying plankton, feces, dead organisms raining down from the surface. But the Ediacaran oceans didn't have plankton, didn't have the massive food webs that exist today. So what were these organisms feeding on? One possibility is microbial mats. For most of Earth's history, the seafloor was covered in bacterial mats, thick layers of photosynthetic cyanobacteria and other microbes producing oxygen and organic carbon. These mats would have been rich in nutrients, and Ediacaran organisms, lying flat on the mat surface, could have absorbed nutrients directly, either from the mat itself or from the water immediately above it, which would have been rich in organic compounds leaking from the bacteria. Another possibility is chemosynthesis. At Mistaken Point, the fossils were living 1,000 meters deep, far below the photic zone where sunlight penetrates. No photosynthesis was possible, but there could have been hydrothermal vents, volcanic fissures on the seafloor releasing hydrogen sulfide, methane, and other reduced chemicals. Modern vent ecosystems support entire communities of organisms that don't rely on sunlight. Instead, they rely on chemosynthetic bacteria that convert chemical energy from the vent fluids into organic carbon. Giant tube worms, clams, shrimp, all living in total darkness, feeding on bacteria. Could Ediacaran organisms have hosted chemosynthetic bacteria? It's possible. Some modern animals, like giant tube worms, have symbiotic bacteria living inside their tissues, providing nutrients. The worm has no mouth, no gut. It's essentially a living tube filled with bacteria. Could Charnia and Fractofusus have been doing something similar, hosting bacteria in their tissues, absorbing the organic compounds the bacteria produced? We don't know. And that's the frustrating part, because the preservation, as good as it is, doesn't give us cellular detail. We can see the external morphology, the branching patterns, the quilted textures. But we can't see inside, can't see if there were bacteria, can't see if there were cells at all. All we have are shapes, patterns, fossils that refuse to fit neatly into any modern category. And then, in the early 2000s, technology caught up, and scientists found a way to peer deeper into these ancient organisms than ever before. A way to test whether they were animals, fungi, or something else entirely. The answer would come from chemistry, specifically from a molecule called cholesterol. Cholesterol is a steroid, a lipid molecule with a very specific structure, 4 fused carbon rings, essential for building cell membranes in animals. All animals produce cholesterol. It's part of what makes animal cells flexible and durable. Plants don't make cholesterol. They make other sterols. Fungi make ergosterol. Bacteria make hopanoids. Each kingdom of life has its own signature lipid molecules, and those molecules, under the right conditions, can survive in the fossil record for hundreds of millions of years. In 2018, a team led by Ilya Bobrovsky from the Australian National University extracted organic molecules from Dickinsonia fossils found in the White Sea region of Russia. Dickinsonia is one of the most famous Ediacaran organisms. It's a flat, oval creature up to 1.5 meters long with a quilted, segmented appearance. It looks like a bath mat or a giant inflatable pool toy. And for decades, scientists argued about what it was. Some said it was a worm, others said it was a cnidarian. Seilacher said it was a vendobiont, something totally unique. Bobrovsky's team used a technique called biomarker analysis. They dissolved small amounts of the fossil in solvents, isolating the organic molecules, and then analyzed them using mass spectrometry. And they found cholesterol. A lot of cholesterol. The lipid signature was unmistakable. Dickinsonia was an animal, not a fungus, not a vendobiont, an animal. The announcement made headlines around the world. The mystery of the Ediacarans, it seemed, was solved. They were animals. Primitive, yes. Weird, definitely. But animals nonetheless. Ancestors, perhaps, of modern animal groups, or at least closely related. But here's the problem. Dickinsonia is just one organism, out of dozens of Ediacaran species. And when scientists tried the same analysis on other fossils, on Charnia, on Fractofusus, on Cyclomedusa, the results were ambiguous. Some had cholesterol, some didn't. Some had mixed signatures, suggesting contamination or unusual biochemistry. The truth is, we still don't know what most Ediacaran organisms were. Some, like Dickinsonia, were probably animals. But others? Maybe they were Vendobionta. Maybe they were early experiments in multicellularity that failed. Maybe they were lichens, symbiotic partnerships between fungi and algae or bacteria. Maybe they were giant protists, single-celled organisms that evolved elaborate structures without ever becoming truly multicellular. Or maybe, just maybe, they were all of the above. A diverse assemblage of unrelated organisms, united only by the fact that they lived in the same geological period and happened to get big at the same time. What we can say with certainty is this. By 555 million years ago, true animals had appeared. And we know this not from Charnia or Dickinsonia, but from a very different kind of fossil. One that shows not just morphology, but behavior. Its name is Kimberella, and it changes everything. Kimberella was discovered in the White Sea region of Russia in the 1990s, though it wasn't recognized as significant until the early 2000s. It's an oval-shaped organism, about 10 centimeters long, with a distinct front and back, bilateral symmetry. Unlike the radial symmetry of Charnia or the quilted pseudosymmetry of Dickinsonia, Kimberella had a clear head and tail, and, more importantly, it moved. We know it moved because of the trace fossils associated with it. Scratch marks, grooves, patterns in the sediment that indicate something was scraping the seafloor, systematically feeding on the microbial mat. Modern animals that feed this way, snails, slugs, some worms, use a radula, a ribbon of tiny teeth, to rasp away at surfaces. Kimberella appears to have had something similar. It moved across the mat, scraping, feeding, leaving a trail behind it. And when it died, it left an impression, not just of its body, but of its behavior. This is the smoking gun. Animals don't just have specific shapes. They do specific things. They move, they feed, they interact with their environment, and Kimberella did all of that, which makes it unambiguously an animal. Possibly the first true bilaterian, the ancestor of the vast majority of animal species alive today, including us. Worms, insects, fish, reptiles, mammals, all bilaterians, all sharing a common ancestor somewhere in the Ediacaran. But Kimberella is rare. Most Ediacaran fossils show no evidence of movement, no trace fossils, no feeding behavior. They just sit there, passive, sessile, like plants, or fungi, or Vendobionta. And then, 541 million years ago, everything changes. The Ediacaran period ends, and the Cambrian begins. And with it, the Cambrian explosion. Within 10 million years, nearly every major animal group appears in the fossil record. Trilobites, brachiopods, mollusks, arthropods, chordates. Suddenly, the oceans are filled with animals that have eyes, claws, jaws, shells, armor, predators and prey, an arms race, a biological revolution, and the Ediacarans. They vanish, not gradually, not slowly replaced by more advanced forms. They just disappear. One moment, the fossil record is full of Charnia and Dickinsonia and Fractofusus. The next moment, they're gone. No transitional forms, no descendants, no evolutionary legacy. It's as if someone flipped a switch and deleted an entire biosphere. What happened? What killed the Ediacarans? And why did animals survive when the Ediacarans didn't? The answer, it turns out, lies in one of the most profound ecological transformations in Earth's history. A change so fundamental that it restructured the entire planet. And it happened in the space of a few million years, right at the boundary between the Ediacaran and the Cambrian. Scientists call it the agronomic revolution, the moment life learned to dig. For 3.5 billion years, from the origin of life until the end of the Ediacaran, the seafloor was a smooth, unbroken surface. Microbial mats covered everything, layer upon layer of bacteria, stabilizing the sediment, creating a living carpet. Nothing disturbed it, nothing burrowed. Life existed on top of the sediment or within the water column, but not inside the sediment itself. The substrate was sterile, inert, a sealed archive. But around 540 million years ago, animals started digging. Not deep at first, just shallow burrows, U-shaped tubes, horizontal tunnels. But it was enough, because once you start churning the sediment, mixing oxygenated water down into the anaerobic muds below, you change everything. The microbial mats, which had dominated the seafloor for billions of years, couldn't survive. They need a stable surface. Once burrowing starts, the mats are ripped apart, destroyed, and with them goes the primary food source for osmotrophic organisms like the Ediacarans. This is called bioturbation, the mixing of sediments by living organisms. And it's so ubiquitous today that we take it for granted. Every ocean floor, every lake bed, every riverbank is churned by worms, clams, crustaceans. The top few centimeters of sediment are a biological mixing zone, constantly overturned, oxygenated, homogenized. But in the Ediacaran, bioturbation didn't exist. The seafloor was laminated, layer upon layer of undisturbed sediment, each layer representing a single year, sometimes a single day. You could read the fossil record like tree rings. Then burrowing animals appeared, and within a few million years, those laminated sediments vanished, replaced by churned structureless mud. The geological signature is unmistakable. It's called the Cambrian Substrate Revolution, and it marks the end of the Ediacaran world. But bioturbation wasn't just bad for microbial mats. It was catastrophic for the Ediacarans themselves, because these organisms, whatever they were, had no defenses, no hard parts, no ability to move quickly or burrow or escape. They just sat there, on the surface, exposed. And when the first predators appeared, animals with jaws and claws and the ability to hunt, the Ediacarans were easy targets, soft, immobile, helpless. We don't have direct evidence of Ediacarans being eaten. Fossilized bite marks are extremely rare, and the Ediacarans, with their quilted construction, probably didn't leave much behind when they were consumed. But the timing is suggestive. The first predators appear in the fossil record right at the Ediacaran-Cambrian boundary, and the Ediacarans disappear at exactly the same time. One of the earliest known predators is a tiny worm called Claudina. It's only a few centimeters long, but it has a hard mineralized tube for a body made of calcium carbonate. And some Claudina fossils show small holes drilled through the tube, predation marks. Something was hunting Claudina, drilling through its shell to get at the soft tissue inside. If predators were going after Claudina, which had armor, they were definitely going after Ediacarans, which had none. So the Ediacarans faced a double catastrophe. Their food source, the microbial mats, was being destroyed by bioturbation, and they themselves were being eaten by the first predators. Caught between starvation and predation, they had no way to adapt. Because adaptation takes time. Evolution through natural selection requires variation, inheritance, and differential survival. But the Cambrian explosion happened too fast. 10 million years is nothing in evolutionary terms. The Ediacarans, whatever they were, couldn't evolve hard parts, couldn't evolve mobility, couldn't evolve predator defenses quickly enough. And so they went extinct, almost all of them. There are a very few possible survivors. Some scientists argue that Charnia-like organisms might have persisted into the early Cambrian. There are controversial reports of Ediacaran-style fossils from Cambrian rocks in Siberia. But the fossils are poorly preserved. The dates are uncertain, and most paleontologists remain skeptical. If Ediacarans did survive into the Cambrian, they didn't last long. The only clear exception is the sponges. Sponges, it turns out, have been around since at least the Ediacaran. Chemical biomarkers for sponges, specific steroid molecules that only sponges produce, have been found in rocks as old as 635 million years. And sponges are still here today, thriving in oceans worldwide. They survived the Cambrian explosion, survived the Ordovician extinction, survived the Permian extinction, the worst mass extinction in Earth's history, survived the asteroid that killed the dinosaurs, survived ice ages and warm periods, and everything else the planet has thrown at them for over 600 million years. Why did sponges survive when the Ediacarans didn't? Because sponges are simple. They don't have organs, don't have tissues really, just loose assemblages of cells. And because they filter feed, pumping water through their bodies and trapping bacteria and organic particles, they weren't dependent on microbial mats. When the mats disappeared, sponges barely noticed. They just kept filtering, kept growing, kept surviving. The lesson is clear. Survival isn't about complexity, it's about flexibility. The Ediacarans, whatever their body plan, were too specialized, too dependent on a specific environment, a specific food source. When that environment changed, they couldn't adapt. But sponges? Sponges are generalists. They can live in almost any marine environment, from tide pools to the deep sea, from the tropics to the poles. They're not impressive. They don't move, don't think, don't do much of anything, but they endure. And that brings us to the final question. What was the legacy of the Ediacarans? If they went extinct, leaving no descendants, why do they matter? The answer is that extinction isn't failure. Extinction is part of evolution. Species go extinct. That's what they do. Over 99% of all species that have ever lived are extinct. What matters isn't whether a lineage survives, but what it teaches the biosphere, what innovations it introduces, what possibilities it opens up. The Ediacarans proved that macroscopic multicellular life was possible. For 3 billion years, life had been microscopic. Bacteria, algae, simple cells floating in the ocean. And then, in the aftermath of Snowball Earth, in a world rich with nutrients and oxygen, life tried something new. It built large bodies. It experimented with different architectures: fronds, quilts, discs, ovals, bilaterians. Some of those experiments worked, others didn't. But the fact that they were tried at all changed everything. Because the genetic toolkit that makes multicellularity possible, the genes that controlled cell adhesion, cell differentiation, pattern formation, Those genes didn't evolve in the Cambrian. They evolved earlier, in the Ediacaran or even before. Hox genes, which control body segmentation in animals, exist in single-celled organisms like chionoflagellates. Pax genes, which control eye development, exist in organisms that don't have eyes. The tools were there in the genome, waiting, and the Ediacarans were among the first to use them. Some Ediacarans, like Dickinsonia and Kimberella, were animals. Primitive animals, yes, but animals nonetheless. And they passed their genes, their body plans, their evolutionary lessons onto the Cambrian fauna. Kimberella might be the ancestor of all bilaterians. Dickinsonia might represent an early branch of the animal tree that went extinct. We don't know. The phylogenetic relationships are still unclear. But what's certain is that animals existed in the Ediacaran, and those animals survived, evolved, diversified, became trilobites, became fish, became us. The Ediacarans that weren't animals, the Vendobionta, if they existed, didn't survive. But they proved something important. They proved that there's more than one way to build a complex organism. Animals used tissues and organs. Vendobionta might have used modular, inflatable chambers. Different solutions to the same problem. How do you get big? How do you stay alive when you're big? How do you gather resources, exchange gases, reproduce? Animals solved those problems one way. Vendobionta might have solved them another. And for tens of millions of years, both solutions worked. But when the environment changed, when the seafloor was overturned and predators appeared, only one solution was flexible enough to survive. The animal solution: mobility, cephalization, predation, the Darwinian arms race. But here's the thing. Just because animals won doesn't mean Vendobionta were inferior. They were adapted to a specific world, the Ediacaran world, a world of calm, mat-covered seafloors, rich in nutrients, free from predation. In that world, Vendobionta thrived. They were successful, but that world ended, and with it, the Vendobionta. Evolution doesn't optimize for survival in all possible futures. It optimizes for survival right now in the current environment. And when the environment changes faster than evolution can adapt, lineages go extinct. It's not a moral judgment. It's just physics, thermodynamics, entropy. The universe doesn't care about your body plan. It only cares whether you can extract energy from your environment efficiently enough to reproduce before you die. The Ediacarans couldn't. Not in the Cambrian world. So they died, and animals took over. And here we are, 600 million years later, the descendants of those early Cambrian survivors, trying to understand what came before. And maybe that's the real legacy of the Ediacarans. They remind us that the animal body plan, our body plan, isn't inevitable. It's not the only solution, it's just the solution that happened to work when the Cambrian explosion occurred. In a different world, under different conditions, Vendobionta might have been the ones to survive, might have been the ones to evolve intelligence, build civilizations, look back across deep time and wonder about the strange animal fossils they found in ancient rocks. But that's not what happened. Animals won. And now we get to tell the story of Earth's first experiment with complex life, the story of the Ediacarans, the organisms that were too strange to understand, too alien to categorize, too beautiful to forget. They lived, they thrived, they went extinct, and in doing so, they paved the way for everything that came after, including us. So let's return to where this story began. Charnwood Forest, Leicestershire, England, 2023. Roger Mason is 81 years old now. He still lives near Leicester, and once a year, weather permitting, he makes the journey back to the quarry, where, as a 15-year-old boy skipping class, he found the fossil that changed paleontology. The fossil is still there, still embedded in the rock face, protected now by law, designated a site of special scientific interest. You can't chip pieces off anymore, can't take samples without permission from Natural England and a research permit. But you can visit. You can stand where Roger stood 66 years ago and run your fingers across the surface of a creature that lived 565 million years ago, when England was part of a volcanic island arc near the South Pole, when the air had no oxygen to breathe, when the only sound was wind and waves and the deep rumble of undersea volcanoes. Roger Mason doesn't talk much about the discovery. He's given a few interviews over the years, appeared in a couple of documentaries, but mostly he's let the fossil speak for itself, which is appropriate, because Charnia masoni, the organism named in his honor, is still teaching us, still revealing secrets. In 2008, a team from the University of Cambridge used laser scanning technology to create three-dimensional digital models of Charnia fossils at Mistaken Point. The resolution was extraordinary. Every branch, every ridge, every subtle variation in texture captured in micron-level detail. And when they analyzed the growth patterns, they discovered something unexpected. Charnia didn't grow continuously. It grew in discrete steps, stages, like a fern unfurling. Each branch would elongate, then stop. A new set of branches would form at the tip, then those would elongate and stop. Modular growth, self-similar at every scale, a fractal. This is not how animals grow. Animals grow through cell division and differentiation. Cells multiply, specialize, form tissues and organs. The process is continuous, coordinated by genetic programs and hormonal signals. But Charnia grew the way crystals grow, or the way fungi grow, by adding modules, repeating units, each identical to the last, but scaled appropriately. It's a completely different strategy. And it suggests that Charnia, whatever it was, wasn't constrained by the same developmental rules that govern animals. It was experimenting, trying different geometries, different architectures, and for tens of millions of years, it worked. Charnia was successful. It colonized seafloors across the globe, from shallow waters to the deep ocean. It reproduced, it thrived, and then it went extinct, but not before leaving its image in stone. A message to the future, a glimpse of what life looked like before the Cambrian explosion, before predators, before the Darwinian arms race that defines modern ecosystems. Charnia is a time capsule, a window into the Ediacaran world. And the more we study it, the stranger that world becomes. Consider this. At Mistaken Point, the fossils are found at a depth of at least 1,000 meters. We know this from the surrounding rocks, from the types of sediments, from the volcanic deposits. This was the deep ocean, and that creates a problem. Because if Charnia was photosynthetic, if it was using sunlight to produce energy like a plant or algae, it couldn't have lived at 1,000 meters. Sunlight doesn't penetrate that deep. The photic zone, where photosynthesis is possible, extends to maybe 200 meters in the clearest ocean water. Below that, it's dark, perpetually dark. So how was Charnia feeding? If not through photosynthesis, Then what? One possibility, as we discussed earlier, is osmotrophy, absorbing dissolved organic carbon directly from the water. But where's that carbon coming from? In modern deep oceans, organic carbon comes from above, from dead plankton and feces and other detritus raining down from the surface. Marine snow, scientists call it. A constant, gentle snowfall of organic particles drifting down through the water column, feeding the deep sea ecosystems. But the Ediacaran oceans didn't have plankton, didn't have the complex food webs that produce marine snow. So the deep ocean should have been a desert, barren, lifeless. And yet Charnia was there, thriving, forming forests, unless there was a different source of carbon, something we haven't considered, something so unexpected that it would rewrite our understanding of Ediacaran ecosystems. In 2019, a team from MIT, led by geobiologist Tanja Bosak, proposed a radical hypothesis. They suggested that the Ediacaran deep ocean wasn't a desert at all. It was fed by chemosynthesis, not photosynthesis, but chemical energy from the Earth itself. Here's how it works. In modern oceans, hydrothermal vents release fluids rich in hydrogen sulfide, methane, and other reduced chemicals. These fluids are hot, sometimes superheated to 400 degrees Celsius, but they cool as they mix with seawater. And in that mixing zone, bacteria thrive. Chemosynthetic bacteria. They use the chemical energy in the vent fluids to convert carbon dioxide into organic carbon, just like photosynthetic organisms do with sunlight. Except no sunlight is required. The energy comes from chemistry, from the Earth's interior. These bacteria form the base of entire ecosystems. Giant tube worms, clams, crabs, fish, all living in total darkness, all ultimately dependent on the chemical energy from the vents. And the bacteria themselves form mats, thick carpets on the seafloor around the vents, producing organic carbon that other organisms can consume. Now, here's the key. The early Earth was volcanically active, much more active than today. The mantle was hotter, the crust thinner, and volcanic activity was widespread. Which means hydrothermal vents were probably far more common in the Ediacaran than they are now. Not just isolated vents along mid-ocean ridges, but vents scattered across the ocean floor, wherever the crust was thin enough for magma to reach the surface. If the deep Ediacaran ocean was dotted with hydrothermal vents, and if those vents supported chemosynthetic bacterial mats, then the deep ocean wouldn't have been a desert. It would have been productive, rich in organic carbon, an oasis. And Charnia, lying flat on the seafloor near these vents, could have absorbed nutrients from the chemosynthetic mats or from the organic compounds dissolved in the vent fluids. This would explain how Charnia survived at depth. But it raises another question. Were the Ediacarans dependent on hydrothermal vents? Or were vents just one of many possible environments they could colonize? The answer seems to be the latter, because Ediacaran fossils are found in a wide range of depositional environments. Deep ocean, yes, but also shallow shelves, coastal lagoons, tidal flats. Some fossils are found in rocks that show evidence of wave action, ripple marks, crossbedding. Others are found in quiet, anoxic basins where the water was still and probably poisonous. The Ediacarans were generalists. They could live almost anywhere, which suggests they weren't dependent on any single food source. So maybe Charnia didn't have one feeding strategy. Maybe it had several. Osmotrophy in shallow water, absorbing organic carbon from microbial mats. chemosynthesis, symbiosis in deep water, hosting bacteria near hydrothermal vents. Maybe even photosymbiosis in sunlit shallows, like modern corals, which host photosynthetic algae in their tissues. The problem is, we can't test these hypotheses. We can't see inside Charnia, can't see if there were bacteria, can't analyze its metabolism. All we have are shapes, external morphology. And shapes, as beautiful and informative as they are, only tell you so much. But there's one more line of evidence, one more clue that might finally tell us what Charnia was. And it comes from the way it reproduced. In 2008, the same Cambridge team that laser scanned the Mistaken Point fossils noticed something odd. The Charnia weren't randomly distributed. They were clustered, groups of large individuals surrounded by halos of smaller ones. And when the researchers measured the spacing, they found a pattern. The small Charnia were always at least a certain distance from the large ones, as if some biological process was preventing them from growing too close. At first, they thought it was competition, large Charnia hogging resources, preventing smaller ones from thriving nearby. But the pattern was too regular, too precise. Competition usually produces messier distributions, with small individuals crushed right up against large ones, fighting for space. Then they considered reproduction. What if the small Charnia were offspring of the large ones? And what if the large ones were releasing spores or seeds or larvae, which drifted a certain distance before settling and growing? That would explain the spacing. It's called a dispersal kernel, the statistical distribution of offspring around a parent. And it's common in plants, fungi, and sessile animals like corals. But here's where it gets interesting. When the researchers measured the dispersal kernel for Charnia, they found two distinct patterns. Some individuals had offspring that settled very close by, within a few centimeters. Others had offspring that settled farther away, up to several meters. Two different reproductive strategies in the same species. This is called dimorphism. And it's common in organisms that have both sexual and asexual reproduction. Asexual reproduction, where an organism clones itself, produces offspring that are genetically identical and settle nearby. Sexual reproduction, where two individuals combine genes, produces offspring that are genetically diverse and often disperse farther, reducing competition with siblings and colonizing new areas. If Charnia had both sexual and asexual reproduction, it was doing something very sophisticated, something that requires coordination, timing, possibly even communication between individuals. This is not simple. This is complex life. But it's still not clear whether Charnia was an animal, because fungi also have sexual and asexual reproduction. So do plants. So do many algae. Reproductive complexity alone doesn't tell us what kingdom Charnia belonged to. So we're stuck. Decades of research, thousands of fossils, cutting-edge technology, and we still can't definitively say what Charnia was. Animal, fungus, alga, or Vendobiont. The Ediacaran's greatest mystery remains unsolved. But maybe that's okay. Maybe the point isn't to categorize Charnia, to force it into one of our modern taxonomic boxes. Maybe the point is to recognize that life in the Ediacaran was genuinely different from life today. That the categories we use, animal, plant, fungus, protist, are modern concepts, products of 600 million years of evolution and extinction and divergence. They don't necessarily apply to the Ediacaran world, because the Ediacaran was a transitional period, a time when life was experimenting, trying out different body plans, different strategies, different ways of being complex. Some of those experiments led to modern groups, others didn't. And Charnia might represent one of the experiments that failed. A body plan that worked for a while, then didn't. A kingdom that thrived, then went extinct. And if that's the case, if Charnia and the other Ediacarans were truly a separate kingdom of life, then their extinction represents something profound. Not just the loss of a few species, but the loss of an entire way of being alive. An entire branch of the tree of life, pruned away by the Cambrian explosion, leaving no descendants, no modern representatives, nothing but fossils. There are only a handful of times in Earth's history when an entire kingdom went extinct. The Ediacarans might be one of them. And that makes their story not just scientifically interesting, but existentially important. Because it tells us that even major branches of the tree of life, even successful, globally distributed groups of organisms, are not immune to extinction. Given the right circumstances, given a changing environment and new competitors and new predators, even kingdoms can die. Which brings us to the present, and to a question that's been lurking beneath the surface of this entire story. What does the Ediacaran extinction tell us about extinctions happening today? About the biodiversity crisis we're living through right now in the 21st century? The parallels are uncomfortable. The Ediacaran extinction was triggered by environmental change, specifically by the oxygenation of the deep ocean and the disruption of microbial mats. Those changes created new niches, new opportunities for animals. But they also destroyed the ecosystems that the Ediacarans depended on. The old world ended, the new world began, and the organisms that couldn't adapt went extinct. Today, we're changing the environment faster than at any point in Earth's history, except during mass extinctions. We're altering the atmosphere, the oceans, the land surface. We're disrupting ecosystems that have been stable for millions of years. And species are going extinct at rates that rival the great mass extinctions of the past. The background extinction rate, the normal rate at which species go extinct due to natural processes, is about 1 species per million per year. Today, we're losing species at a rate 100 to 1,000 times higher. Some estimates suggest we're in the early stages of a 6th mass extinction. And just like the Ediacaran extinction, the species going extinct today are often the specialists, the ones adapted to specific environments, specific food sources, specific ecological niches: pandas, rhinoceroses, coral reefs, rainforest species. They're like the Ediacarans, successful in their world, thriving even, but unable to adapt quickly enough when that world changes. The generalists, the ones with flexible diets and broad tolerances, are doing fine. Rats, pigeons, cockroaches, dandelions, they're the sponges of the modern world. Unremarkable, not impressive, but enduring. Does that mean we should only care about preserving generalists? That specialists are doomed anyway, so why bother? No, absolutely not. Because the loss of specialists is the loss of diversity. And diversity is what makes ecosystems resilient. An ecosystem with 100 species, each specialized for a different niche, is far more stable than an ecosystem with 10 generalists all doing the same thing. Diversity provides options, redundancy, insurance against catastrophe. The Ediacaran world had diversity, dozens of species, each with a different morphology, a different strategy. And when the environment changed, most of them went extinct, but a few, The flexible ones, the ones that happened to have traits that worked in the New World, survived. And those survivors became the ancestors of all modern animals. If the Ediacaran extinction had been total, if every single Ediacaran organism had gone extinct with no survivors, there would be no Cambrian explosion, no animals, no fish, no dinosaurs, no mammals, no us. The survival of a few flexible Ediacaran lineages, the ancestors of Kimberella and the other early bilaterians, was essential for everything that came after. So diversity matters. Not just because it's beautiful, though it is. Not just because it's scientifically interesting, though it is. But because it's the raw material of future evolution. The more diversity we preserve today, the more options life has to adapt to the changes coming tomorrow. And the changes are coming. Climate change, ocean acidification, habitat destruction, pollution. These aren't theoretical future problems. They're happening now. And species are responding the same way the Ediacarans did. Some are adapting, shifting their ranges, changing their behaviors, evolving. Others aren't. They're going extinct. We don't get to choose which species survive. Evolution makes that choice. But we do get to choose how much diversity is available for evolution to work with. And right now, we're reducing diversity. Rapidly. We're creating a world with fewer specialists, fewer niches, fewer options. A world more like the early Cambrian, where a handful of generalist survivors rebuild ecosystems from scratch. That rebuilding takes millions of years. After the Permian extinction, the worst mass extinction in Earth's history, it took 10 million years for ecosystems to recover. 10 million years of impoverished biodiversity, simplified food webs, empty niches. Eventually, life bounced back. It always does. But the recovery time is measured in geological epochs, not human lifetimes. We won't be around to see it. So the lesson from the Ediacarans isn't just about deep time. It's about now. It's about recognizing that the extinctions we're causing today will echo through the future in ways we can barely imagine. The species we're losing aren't just data points. They're lineages, evolutionary experiments. Each one represents millions of years of adaptation, refinement, survival. And when they go extinct, that entire history is erased, gone, unrecoverable. The Ediacarans went extinct 541 million years ago, and we're still trying to understand what they were, still piecing together their story from fragments of stone, still wondering what the world looked like before the Cambrian explosion. 600 million years from now, if intelligence evolves again, if some distant descendant of rats or crows or octopuses develops science and starts studying the fossil record, they'll find our layer, the Anthropocene. They'll see a sudden spike in extinction rates, a dramatic shift in species composition, a change in the geological record as profound as the Ediacaran-Cambrian boundary. And they'll wonder, what happened? What were those lost species? What did the world look like before the change? And maybe, if we're lucky, they'll find fossils, impressions of elephants and tigers and pandas embedded in stone, and they'll study them the way we study Charnia. They'll argue about what they were, how they lived, why they went extinct. They'll use advanced technology we can't imagine to analyze the fossils, to extract chemical signatures, to reconstruct ecologies. And they'll conclude, probably, that these organisms were specialists, adapted to specific environments that no longer exist, beautiful, complex, successful in their time, but unable to adapt when the world changed. and they'll be right, unless we change course, unless we recognize that diversity matters, that extinctions are forever, that the choices we make today will determine what the world looks like for millions of years to come. The Ediacarans didn't have a choice. They went extinct because the world changed and they couldn't adapt fast enough. But we have a choice. We're causing the changes, which means we can stop them, or at least slow them down. Give species time to adapt, preserve habitats, reduce emissions, protect ecosystems. It's not too late. Not yet. The sixth mass extinction is just beginning. We're in the early stages. The window is still open, but it's closing. The Ediacarans teach us that even globally successful organisms can go extinct if the environment changes too quickly. But they also teach us that survival is possible. That a few flexible lineages can endure, adapt, and ultimately give rise to new forms of life, new diversity, new possibilities. We're not Ediacarans. We're not sessile organisms, passively absorbing nutrients, unable to move or adapt. We're animals, descendants of the survivors, products of 600 million years of evolutionary arms races, adaptations, and innovations. We have brains, tools, technology, the ability to understand what's happening and to do something about it. The question is, will we? Or will we be like the Ediacarans, successful in our time, but unable to adapt when the world changes, leaving only fossils for some future intelligence to puzzle over, wondering what we were and why we disappeared? The choice is ours, and the clock is ticking. But enough about the present. This is a story about the past, about deep time, about the Ediacaran world and the creatures that inhabited it. So let's go back, one more time, to the seafloor 560 million years ago, to the forests of Charnia and Fractophusus swaying in gentle currents, to the microbial mats glowing faintly with bioluminescence in the darkness, to a world so alien, so unlike anything we know that it might as well be another planet. Imagine you're there, floating in the water column, a kilometer below the surface. It's dark, not the blackness of a moonless night, but the absolute total darkness of the deep ocean. No light penetrates here, no stars, no sun, just darkness. But your eyes adjust, or rather, your instruments adjust. Because if you were actually there, you'd be crushed by the pressure. 100 atmospheres, enough to collapse your lungs, rupture your cells, kill you instantly. But let's pretend you're in a submersible, a vessel strong enough to withstand the pressure, with lights to pierce the darkness. You descend. The water is cold, just above freezing, and it's quiet. No sound but the hum of your engines and the occasional creak of the hull as pressure fluctuates. The water is clear, clearer than modern oceans, because there's no plankton, no detritus, no marine snow, just water and dissolved minerals and organic carbon leaking from the seafloor below. Then you see the bottom, a flat, featureless plain of mud, grey, unbroken, and covering the mud, a thin film of bacteria, a microbial mat. It glows, faintly. Bioluminescence, produced by chemosynthetic bacteria processing sulfur compounds from the sediment. The glow is blue-green, ghostly, barely perceptible, but it's enough to illuminate the landscape. And rising from the mat, like trees in a forest, are the Charnia, hundreds of them, thousands, each one a delicate frond, branching and rebranching in fractal patterns, swaying slightly in a current you can't feel. They're beautiful, alien, utterly silent. No movement except the gentle wave of the current. No feeding, no hunting, No interaction, just existence. Passive, eternal. You drift closer. Examine one of the larger specimens. It's 60 centimeters tall. The branches are flattened like ribbons, each one a few millimeters wide. The surface is smooth. No texture, no cells visible to the naked eye. Just a uniform, quilted appearance. The color is pale, whitish, with hints of pink or orange from carotenoid pigments, the same pigments that color modern algae and bacteria. At the base, a disc, a holdfast, anchoring the organism to the mat, but not penetrating it, just sitting on top like a sticker on a surface. You reach out with the submersible's manipulator arm, touch the frond. It's soft, flexible, not rigid like a plant stem, more like a jellyfish, but not quite. There's structure there, internal chambers. You can feel them, inflated, pressurized, giving the organism shape without a skeleton. You pull back, drift over the mat, and you see more, different species. Fractophusus, with its chaotic branching, looking like a fern that grew wrong. Bradgartia, tall and feathery. Pectinifrons, wide and fan-like. And scattered among them on the mat surface are the flat ones. Dickinsonia, ovals, quilted, up to a meter across, lying completely flat, absorbing nutrients, doing nothing. It's peaceful, serene. This is a world without predators, without competition, without violence. Organisms grow, reproduce, die, decompose, and their nutrients are recycled by the mat, feeding the next generation. It's a balanced ecosystem, stable, sustainable. It's been this way for tens of millions of years, and it will continue forever, except it won't. Because 1,000 kilometers away in shallow water near the coast, something new is happening. Animals are evolving. Kimberella is scraping the mat, feeding, moving, leaving trails. And those trails are the beginning of something. The beginning of bioturbation, of burrowing, of disruption. It will take millions of years. But eventually those burrowing animals will spread. They'll move into deeper water. And when they do, the mats will start to disappear. The sea floor will be churned, oxygenated, and the Charnia forests will die. Not because the animals hunt them, not because they compete directly, but because the world they depend on, the mat-covered seafloor, will cease to exist. And in the shallow water, predators are evolving. Claudina, with its hard shell, is just the beginning. Soon there will be Anomalocaris, a meter-long apex predator with compound eyes and grasping appendages. There will be trilobites, scuttling across the seafloor, feeding on anything soft and slow. There will be jaws, claws, armor, the Cambrian explosion, and the Ediacarans, beautiful and passive and defenseless, will go extinct. But not yet. Right now, in this moment, 560 million years ago, they're still here, still thriving, still swaying in the current, unaware that their world is ending. Unaware that in a few million years, they'll be gone, replaced by something faster, smarter, more aggressive. But for now, they're alive, and that's enough. You rise, leave the seafloor, ascend through the darkness. The Charnia forests fade below you, disappear into the black, and you surface, break through the water into the air, an air that's toxic, methane, carbon dioxide, almost no oxygen. You can't breathe it. You die in seconds. Above you, the sky is red. A young sun, dimmer than today, barely above the horizon. The moon, huge, 3 times the size it appears now because it's closer. Orbiting just 200,000 kilometers away, not the 380,000 it's at today. Tides are extreme. The day is short, 21 hours, the planet spins faster, and on the distant shore, you see volcanoes, dozens of them, erupting, ash clouds rising, lava flows glowing orange against the red sky. This is a violent planet, a young planet, still settling, still cooling, still figuring out how to be a world. But it's alive. For the first time in Earth's history, complex life covers the seafloor. Not just bacteria, not just algae, but organisms you can see, touch, study, the Ediacarans, and they're teaching us, across the immense gulf of time, across 560 million years. They're teaching us that life is resilient, that it experiments, that it tries different solutions, different strategies, different ways of being complex. Some of those strategies work, some don't. But the trying is what matters, because without the Ediacaran experiments, there would be no Cambrian explosion. Without the failures, there would be no successes. Evolution learns from extinction, and the lessons learned in the Ediacaran shaped everything that came after, including us. We are the descendants of the survivors, of Kimberella and the early bilaterians, of the organisms that adapted when the world changed, that evolved mobility, predation, defense. That survived the Cambrian explosion and went on to colonize every niche, every habitat, every corner of the planet. But we're also inheritors of the Ediacaran legacy, the genetic toolkit that makes multicellularity possible, the developmental programs that control body patterning, the cell adhesion molecules, the regulatory networks. Those tools didn't evolve in the Cambrian, they evolved earlier, and the Ediacarans were among the first to use them. So when you look at a human body, at its segmentation, its bilateral symmetry, its complex tissues and organs, you're looking at innovations that have roots in the Ediacaran. We're not descended from Charnia, but we're part of the same story, the same evolutionary narrative. The story of life learning to be complex, to be big, to be diverse. And that story is still being written. Because evolution doesn't stop. It's happening right now, today. In oceans and forests and cities, species are adapting, evolving, responding to the changes we're causing. Some will survive, some won't. But the process continues. And 600 million years from now, if there's anyone around to study the fossils, they'll look back at our time and see another transition, another boundary, the Anthropocene. They'll see extinctions. They'll see species disappearing, but they'll also see survivors, adapters, the ancestors of whatever comes next. And maybe they'll wonder, the way we wonder about the Ediacarans, what were those organisms? Why did they go extinct? What did the world look like before the change? And the answer, as always, will be complicated, nuanced, because life is messy. Evolution doesn't follow rules. It doesn't optimize. It just responds to environment, to competition, to chance. And the organisms that happen to have the right traits, the right adaptations, the right luck survive. The others don't. The Ediacarans didn't have the right traits, not for the Cambrian world, so they went extinct. But they weren't failures. They were pioneers, the first to prove that complex life was possible, the first to build large bodies, to colonize the seafloor, to create ecosystems. They succeeded for tens of millions of years, and then they didn't. But their legacy endures, in the fossils, in the genes, in the evolutionary lessons learned, and in the reminder that even successful organisms, even globally distributed groups, are not immune to extinction. Adaptability is survival, specialization is risk. And in a changing world, the flexible inherit the earth. That's the lesson from the Ediacarans. That's what they teach us across the gulf of deep time. Not through words, but through stone, through fossils, through the silent testimony of organisms that lived and died 500 million years ago, leaving only impressions, only shadows, only questions. Were they the first animals? Maybe. Some of them. Probably Dickinsonia. Probably Kimberella. But others? Probably not. Probably something else. Something we don't have a name for. Something that doesn't fit our categories. And maybe that's the most important lesson of all. That nature doesn't care about our categories. Doesn't care about our need to classify, to organize, to put things in boxes. Nature experiments. Tries everything. And most experiments fail. But the ones that succeed, the ones that happen to work, shape the future. The Ediacarans were an experiment, a test, Earth's first try at complex life. And it was beautiful, alien, strange, successful, for a time. And then it ended, and something new began. The Cambrian explosion, the age of animals, our age. But the Ediacarans came first. They paved the way, proved it was possible, and for that we remember them, honor them, study them, because they were the pioneers, Earth's first giants, and their story, as incomplete as it is, as full of mysteries and unanswered questions, is one of the most important stories in the history of life. The story of what came before. The story of the Ediacaran world. The story of organisms too strange to understand, too alien to categorize, too beautiful to forget. They lived, they thrived, they went extinct, and in doing so, they made everything else possible. But there's one more chapter to this story, one more piece of the Ediacaran puzzle that we haven't fully explored. And it's perhaps the strangest piece of all, because it involves an organism that challenges every assumption we've made about what the Ediacarans were. An organism so bizarre, so geometrically impossible by modern standards, that when it was first discovered, scientists thought it had to be a mineral formation, a crystal, not alive at all. Its name is Tribrachidium, and it might be the key to understanding the entire Ediacaran world. Tribrachidium was discovered by Reg Sprigg during his original surveys in the Flinders Ranges in the 1940s. But like so many of his discoveries, it was ignored for decades, dismissed as poorly preserved, misidentified, not taken seriously. It wasn't until the 1980s, when better specimens were found in Namibia and Russia, that paleontologists started paying attention. And what they saw defied explanation. Tribrachidium is small, about 2 centimeters across, disc-shaped. But unlike other disc-shaped Ediacarans, which have radial symmetry or quilted patterns, Tribrachidium has 3-fold rotational symmetry, triradial. 3 arms, spiraling out from a central point, each arm identical to the others, but rotated 120 degrees, like a biological triskelion, a 3-bladed pinwheel frozen in stone. 3-fold symmetry doesn't exist in modern animals, not in any major group. Animals have 2-fold symmetry, like us, bilateral, with a left and right side, or they have 5-fold symmetry, like starfish and sea urchins, pentaradial. Or they have 6-fold, 8-fold, or higher order radial symmetry, like some jellyfish and corals. But never 3-fold. Never. Why? Because 3-fold symmetry is geometrically awkward. It doesn't divide evenly. It creates problems with body patterning, with development, with the placement of organs. If you're a bilateral animal, you can have a gut that runs from front to back, with a mouth at one end and an anus at the other. If you're pentaradial, you can have 5 sets of organs, evenly spaced around a central axis. But if you're triradial, where do you put the gut? How do you organize the body? It's a geometric nightmare. And yet, Tribrachidium did it, for millions of years, successfully. Which suggests that Tribrachidium wasn't constrained by the same developmental rules that govern animals. It was using a different toolkit, a different set of genetic instructions, something we don't see in modern organisms. In 2012, a team from the University of Adelaide, led by paleontologist Diego Garcia Bellido, used high-resolution CT scanning to examine Tribrachidium fossils from South Australia. They were looking for internal structures, anything that might reveal how this organism was put together. And they found something unexpected. The 3 arms weren't solid. They were hollow, inflated, like tubes. And the tubes didn't radiate straight out from the center. They spiraled, coiled around each other in a complex 3-dimensional pattern that looked, in cross-section, like a Celtic knot, overlapping, interwoven, creating a structure that was both beautiful and utterly alien. Garcia Bellido proposed that Tribrachidium was a filter feeder, that water flowed into the center through the spiraling tubes and out through pores in the surface, trapping particles along the way, like a sponge, but more complex, more organized, with directional flow and controlled filtration. If true, this would make Tribrachidium the earliest known filter feeder, predating sponges, predating any Cambrian filter-feeding animals. It would mean that the Ediacarans weren't just passive osmotrophs, lying on the seafloor absorbing nutrients. Some of them were actively pumping water, processing it, extracting food, doing things we associate with animals. But here's the problem. Filter feeding requires muscles, or cilia, or flagella, some way to move water, and there's no evidence that Tribrachidium had any of those. The fossil shows structure, yes, tubes and chambers and pores, but no muscle tissue, no cell layers that could contract, nothing that could pump, unless it didn't need to pump, unless water moved through Tribrachidium passively, driven by currents. The organism oriented itself correctly, the tubes directed flow, and particles were trapped without any active effort, like a carefully designed filter positioned in a stream. This is speculation, educated guessing based on morphology and modern analogues. But it's the best we have. Because Tribrachidium, like so many Ediacarans, doesn't fit neatly into any category. It has features that suggest an animal, filter feeding, directional flow, complex 3-dimensional structure. But it also has features that suggest something else. Modular construction, inflated chambers, 3-fold symmetry. It's a chimera, a hybrid, or maybe just something entirely new. And that's the point. The Ediacaran world was full of organisms like Tribrachidium, organisms that tried body plans we never see today, geometries that don't work in the modern world, strategies that were successful for a time, then abandoned, evolution experimenting, testing the limits of what's possible. 3-fold symmetry was possible for Tribrachidium, but it didn't lead anywhere. No modern animals have it. No descendants survive to pass it on. The experiment ended, and bilateral and radial symmetry won, because they're more flexible, more adaptable, better suited for the Cambrian world of predators and mobility and competition. But the fact that 3-fold symmetry existed at all tells us something important. It tells us that early in the history of complex life, before the rules were set, Before development was locked into specific pathways, almost anything was possible. Life was experimenting with forms that would later be discarded, body plans that would never be tried again. Tribrachidium is a window into that experimental phase, a glimpse of what evolution was like when it was young, before it had learned what worked and what didn't, before it had settled on the designs that would dominate for the next 600 million years. And there are others, other Ediacarans with impossible geometries, organisms that challenge our understanding of what life can be. Take Pteridinium, another Namibian fossil. It looks like a feather or a frond. But when you examine it closely, you realize it's made of 3 overlapping blades spiraling around a central axis. And the blades aren't attached at the base. They're free-floating, held together only by the sediment that buried them. How did this organism maintain its shape when it was alive? How did the blades stay together? No one knows. Or take Ernietta, a sac-like organism, oval, inflated, with internal chambers that look like honeycomb. It sat on the seafloor, half buried in sediment. No mouth, no opening, completely sealed. How did it feed? How did it exchange gases? Again, no one knows. The best guess is osmotrophy, absorbing nutrients through the skin. But the surface-to-volume ratio is terrible. Ernietta is too large, too thick for simple diffusion to work efficiently. It should have suffocated, but it didn't. It thrived for millions of years. Or take Swartpuntia, named after Swartpunt, a farm in Namibia where the fossils were found. It's a branching structure, like Charnia. But instead of fractal branching, where each branch splits into smaller identical branches, Swartpuntia has chaotic branching. Random. No pattern. No self-similarity. It looks like someone spilled paint and let it dry. Organic, biological, but utterly without structure. How did it grow? Was it a colony of individuals, each growing independently? Or a single organism with no developmental program, no genetic control over its shape? We don't know, and the fossil doesn't tell us. It's just there, a tangle of branches, proof that something lived and grew, but no explanation for how or why. These organisms, Tribrachidium, Pteridinium, Ernietta, Swartpuntia, they're the Ediacaran's deepest mysteries. They're not the famous ones, not Charnia or Dickinsonia, which get all the attention. They're the weird ones, the outliers, the organisms that don't fit any hypothesis, that resist explanation. And maybe that's the lesson. Maybe the Ediacaran world wasn't meant to be understood. Not fully. Not by us. Because we're looking at it through the lens of modern biology, through categories and classifications that were shaped by 600 million years of evolution. We're asking questions like, is it an animal? Or how did it feed? Or what's its closest modern relative? But those questions assume that the Ediacaran world operated by the same rules as the modern world. And maybe it didn't. Maybe the Ediacaran was genuinely alien, a different experiment, a different way for life to be complex. And when that experiment ended, when the Cambrian explosion began, the rules changed. The old body plans stopped working. The old strategies failed. And life had to start over, build new forms, new relationships, new ecosystems. The Cambrian explosion is often portrayed as a creative event, a burst of diversity, the origin of modern animal phyla. And it was. But it was also a destructive event, a filter, a bottleneck. It eliminated forms that had been successful for millions of years. replaced them with new forms that were better adapted to a world of predation and competition. The Ediacarans were casualties of that transition. Victims of progress. They didn't fail because they were inferior. They failed because the world changed, and change in evolution is often fatal, but their fossils remain, scattered across the planet, in Australia, Namibia, Russia, England, Canada, China. Everywhere geologists have looked for late Precambrian rocks, they found Ediacarans. And every new discovery adds another piece to the puzzle. Another data point, another mystery. In 2016, a team from the Chinese Academy of Sciences announced the discovery of Ediacaran fossils in southern China, in rocks dated to 550 million years ago, just 9 million years before the Cambrian boundary. These fossils were different from anything found before, small, delicate, with fine, detailed structures preserved in 3 dimensions. And among them were organisms that looked, for the first time, unambiguously like animals. Not weird animals, not ambiguous animals, but recognizable animals, with mouths, with guts, with segmented bodies and appendages. These were the precursors, the ancestors, the organisms that would survive the Ediacaran extinction and give rise to the Cambrian fauna. And they were living alongside the classic Ediacarans, Charnia, Dickinsonia, Tribrachidium, all in the same rocks, the same ecosystems, which means that for at least a few million years, at the very end of the Ediacaran, 2 worlds coexisted. The old world of passive, sessile, osmotrophic organisms, and the new world of active, mobile, predatory animals. And then, the old world ended, suddenly, catastrophically, leaving the new world to inherit the earth. This is the pattern we see throughout the history of life. Old forms persist until they can't, and new forms wait in the wings, ready to take over when the opportunity arises. Dinosaurs dominated for 160 million years, until an asteroid gave mammals their chance. Trilobites ruled the Paleozoic oceans for 300 million years, until the Permian extinction cleared the board. And the Ediacarans, they dominated for maybe 70 million years, until the Cambrian explosion, 70 million years. That's longer than the entire age of mammals, from the extinction of the dinosaurs to today. Longer than the age of flowering plants. Longer than most successful groups of organisms last. The Ediacarans weren't a brief experiment. They were a major chapter in the history of life. And their extinction wasn't a minor event. It was a mass extinction. One of the big ones. But because it happened so long ago, because the fossils are so hard to interpret, because we're only beginning to understand what the Ediacarans were, we don't think of it that way. We don't call it the Ediacaran extinction. We call it the Cambrian explosion. We focus on what appeared, not what disappeared. On the winners, not the losers. But the losers matter, because they show us that success in evolution is temporary, that dominance is fleeting, that no matter how well adapted you are, no matter how successful, no matter how long you've been around, you're vulnerable. The environment will change, and when it does, you either adapt or go extinct. The Ediacarans couldn't adapt, not fast enough, and so they went extinct. But they left us a gift, the gift of their fossils, the gift of their story, a reminder that life is resilient, but not invincible, creative, but not infinite, diverse, but not immune to catastrophe, and a reminder that the world we live in today, the world of animals and plants and fungi, of predators and prey and ecosystems structured by competition isn't the only way life can be. It's just the way that survived, the experiment that worked, for now. But evolution isn't done. It's still experimenting, still trying new forms, new strategies, new ways of being alive. And someday, millions of years from now, the world will change again. And the organisms that dominate today, the mammals, the birds, the insects, the flowering plants, they'll face the same test the Ediacarans faced. Adapt or die. Some will adapt, some won't. And future paleontologists, whatever species they are, will look back at our time and see another boundary, another extinction, another transition from the world that was to the world that will be. And they'll ask the same questions we're asking about the Ediacarans. What were they? How did they live? Why did they go extinct? And the answers, as always, will be incomplete, because the past is hard to read. The fossil record is fragmentary, and the organisms themselves are gone, leaving only impressions, only shadows, only questions. But that's okay, because the questions are what drive science. The mysteries are what make paleontology exciting. And the fossils, imperfect as they are enough. Enough to tell us that there was once a world very different from ours. A world of Charnia forests and Dickinsonia mats and Tribrachidium spirals. A world without predators, without fear, without the Darwinian struggle that defines modern life. That world existed for 70 million years, and then it ended. And we're left with the fossils, the stories, the lessons, and the wonder. The sheer wonder of knowing that life, creative, resilient, endlessly inventive life, tried something completely different, built organisms that have no modern equivalents, created ecosystems that operated by different rules, and thrived until it couldn't. That's the story of the Ediacarans. That's their legacy. Not descendants, not evolutionary lineages that survive to the present, but lessons. Proof that life is more diverse, more experimental, more surprising than we ever imagined. Proof that the categories we use, animal, plant, fungus, are modern conveniences, products of recent evolution, not universal truths. And proof that extinction, as tragic as it is, as final as it is, isn't failure. It's part of the process, part of the story. The Ediacarans went extinct, but they paved the way for animals, for us. And without them, without their experiments, without their pioneering efforts to be complex in a simple world, we wouldn't be here. So we honor them, study them, try to understand them, knowing we'll probably never fully succeed, because they were too different, too alien, too far removed from anything we know. But that's what makes them beautiful. That's what makes them important. They were Earth's first giants, our planet's first experiment with macroscopic complexity. And they were magnificent, strange, impossible, magnificent, and they were real. 560 million years ago, if you could have stood on that seafloor in the darkness, surrounded by forests of Charnia and fields of Dickinsonia, you would have witnessed something no human has ever seen. A world before animals, before predators, before the struggle for survival became violent. A world of quiet growth and slow reproduction and nutrient cycling. A world at peace. That peace didn't last. It couldn't. Because evolution doesn't stop. Competition doesn't end. And eventually, organisms that were faster, smarter, more aggressive arose. And the peaceful world ended, replaced by our world, the world of tooth and claw, of predator and prey, of the Darwinian arms race. But for 70 million years, there was another way, the Ediacaran way. And we're lucky, incredibly lucky, that the fossils survived, that we found them, that we're able, however imperfectly, to reconstruct that lost world. Because it tells us something we need to hear, something important, something hopeful, that life is more than competition, more than violence, more than the struggle to survive. Life is also cooperation, symbiosis, peaceful coexistence. The Ediacarans proved that complex organisms can thrive without predation, without aggression, without the need to kill or be killed. They didn't survive. But they existed for a long time, and that's enough. Enough to show us that the world doesn't have to be the way it is. That evolution could have taken a different path, that the Cambrian explosion, with all its violence and innovation and diversity, wasn't inevitable. It was contingent, a product of specific circumstances, specific changes. In another world, on another planet, under different conditions, maybe the Ediacarans would have survived. Maybe they would have continued evolving, developing new forms, new strategies. Maybe they would have become intelligent, built civilizations, looked up at the stars and wondered if they were alone. But that's not what happened. Not here. Not on Earth. Here, the Cambrian explosion happened. The animals took over, and the Ediacarans went extinct. But we remember them, and we learn from them, and we carry their story forward. Because that's what science does. It recovers the past, honors it, learns from it, and uses those lessons to understand the present and imagine the future. The Ediacarans are gone, but their fossils remain, embedded in stone, waiting to be found, waiting to teach us, waiting to remind us that the history of life is longer, stranger, and more wonderful than we ever imagined. And the story isn't over. It's still being written by us, by every species on Earth, by evolution itself. The Ediacaran was one chapter, the Cambrian was another, the Cenozoic is another, and the Anthropocene, our chapter, is just beginning. What will it say? What will our fossils tell future paleontologists, if any exist? Will they see diversity or extinction, innovation or destruction, cooperation or competition? The answer depends on what we do, on the choices we make, on whether we learn the lessons the Ediacarans are trying to teach us. That survival isn't guaranteed, that dominance is temporary, that even successful organisms, thriving ecosystems, entire kingdoms of life can go extinct if the environment changes too fast. But also that life is resilient, that extinction isn't the end, that from the ashes of the old, new forms arise, new possibilities, new chapters. The Ediacarans went extinct, but they made the Cambrian possible, and the Cambrian made everything else possible, including us. So we're not the end of the story. We're just another chapter, another experiment, another attempt by evolution to figure out what works and what doesn't. And maybe, millions of years from now, when we're fossils, some future intelligence will look back at us the way we look at the Ediacarans, with wonder, with curiosity, with questions. What were they? How did they live? Why did they go extinct? And maybe they'll learn the same lessons we're learning, that life is precious, that diversity matters, that extinction is forever, and that the choices we make today echo through deep time in ways we can't predict. The Ediacarans didn't get to choose. The world changed and they went extinct. But we have a choice. We're causing the changes, which means we can control them, slow them, stop them, give life time to adapt, time to experiment, time to find new solutions. The Ediacaran world ended, but its legacy endures. In the fossils, in the genes, in the lessons learned, and in the reminder that life, creative, resilient, endlessly inventive life, always finds a way, even after extinction, even after catastrophe, even after the world ends. Because the world doesn't end, it changes. And change, as destructive as it is, as painful as it is also opportunity. Opportunity for new forms, new experiments, new chapters. The Ediacarans were Earth's first experiment with complex life, and they were magnificent. And they failed. And that's okay, because failure in evolution is how we learn, how we improve, how we move forward. The Cambrian explosion succeeded because the Ediacaran extinction failed. The age of animals succeeded because the age of Vendobionta ended. And whatever comes next will succeed because our age, the age of humans, will eventually end too. That's not pessimism. That's realism. That's deep time. That's evolution. Nothing lasts forever, but everything leaves a mark. The Ediacarans left their fossils. We'll leave ours. And those fossils will tell stories. Stories of what we were, what we did, what we created, what we destroyed. And maybe, just maybe, those stories will teach future life the same lessons the Ediacarans are teaching us. That diversity is strength. That adaptation is survival. That extinction is a teacher. And that life, no matter how many times it falls, always gets back up, always tries again, always experiments, always creates. Because that's what life does. It lives, it thrives, it evolves, it goes extinct, and it starts over. Again, and again, and again. For 4 billion years, and counting. The Ediacarans were part of that story. A beautiful, strange, impossible part. And their chapter, as short as it was, as tragic as its ending was, matters. Because it shows us what's possible, what life can be, what forms it can take, what worlds it can create. And it reminds us that we're part of the same story, the same process, the same evolutionary experiment. We're not separate from nature. We're not above it. We're in it, part of it, products of the same forces that created Charnia and Dickinsonia and Tribrachidium. And someday we'll be fossils too, impressions in stone, shadows in sediment, stories for some future intelligence to piece together. But until then, we're alive, and we have a chance to learn, to adapt, to change, to make choices that honor the past and protect the future. The Ediacarans didn't get that chance. The world changed too fast. They couldn't adapt. They went extinct. But we can. We have the knowledge, the technology, the awareness. We know what's happening. We know what's at stake. And we know from 4 billion years of evolutionary history that diversity is the key, that flexibility is survival, that the more options life has, the better its chances. So let's preserve those options, protect that diversity, give life in all its forms the chance to continue, to experiment, to evolve, to create new wonders we can't imagine. Because the story of life isn't over. The Ediacaran was one chapter, the Cambrian was another, the Cenozoic is another, and the next chapter, whatever it is, whoever writes it, will be built on the foundation we leave behind. Let's make it a good one. Let's honor the Ediacarans, not just by studying their fossils, but by learning their lessons, by recognizing that we're part of a continuum, a story that began 4 billion years ago and will continue long after we're gone. The Ediacarans were Earth's first giants, the pioneers of complex life, the proof that life can be more than we imagine, more diverse, more creative, more beautiful. They lived, they thrived, they went extinct, and in doing so, they made everything else possible, including us, including this moment, including the ability to look back across 560 million years and see, in fossils embedded in stone, a world that once was, a world that will never be again, but a world that matters, because it was real, because it existed, because it tried, and that's enough. That's more than enough. That's everything.